There we go. Topic 1, inheritance. Okay. If you think back to doing grade 10 genetics way back when, you would have come across these things called Punnett squares and pedigree charts. Essentially, this topic is just a revision of those two concepts and extending your knowledge on them a little bit. And so I'm just going to go through a basic overview of genetics, and then we'll get into all of the inheritance patterns. So a couple things to go over first. The first thing that you guys need to know is what an allele is. So within the human genome, every single one of our cells has 23 pairs of chromosomes. 23 pairs of chromosomes, or 46 in total. If you've seen a picture of a karyotype, you would know what they look like, but essentially, this could be chromosome number 1, this could be chromosome number 2 of the 46, and then the pair of them together is pair number 1 of the 23. One of these chromosomes is a maternal chromosome, so you've inherited it from your mother. The second chromosome is inherited from your father. And the way that it works is each chromosome in the pair is the same version of each chromosome. It's the same version in the sense that every single gene is located in the same spot. For example, this gene that is highlighted here, gene number B, could be the gene for, I don't know, hair color, as an example. And this is the hair color gene that you've inherited from your mother. The hair color gene that you inherit from your father is on the exact same spot and position as the corresponding chromosome. And basically, even though these genes code for a particular trait, there are different variations of the same gene, which are called alleles. So alleles are variations of the same gene. Hopefully, this is revision from grade 10. Same gene. And if we're looking at the basics, they can either be dominant or recessive. And basically, the way that that works is if you inherit a copy of the dominant gene from your parents, that is the gene that gets expressed physically. The recessive gene will only be expressed if you inherit 2 copies of a recessive gene from your parents. The combination of alleles that you may have make up your genotype. So genotype is the combination of alleles and are denoted by letters, basically. So in this case, the gene for hair color, I am marking it by the letter B. Capital stands for the dominant gene. The lowercase b stands for the recessive gene. If you've inherited 2 copies of the same gene, for example, here and here, both of them are the same letter, we say that the genotype is homozygous, this one being homozygous dominant, and this one being homozygous recessive. And if you've inherited a copy of 2 different types of genes, so 1 copy of the dom and 1 copy of the recessive, you are heterozygous for the gene. The alleles making up the genotype will then influence what is known as the phenotype. So this is actually the physical representation of these genes, or the physical trait that the genes code for. Code for. So for example, if you've got a genotype of capital B, where the dominant gene codes for black hair and the recessive gene codes for blonde hair. These are completely fake examples, by the way. Blonde hair. If you've got this is a homozygous dominant genotype, but the phenotype is black hair. Similarly, if you've got capital B, little b as your genotype, even though you do have that recessive allele, the dominant one is the one that's expressed in the phenotype, your phenotype is still going to be black hair. And so all of our inheritance patterns that we're going to be looking at deal with how genes are passed on between parents and their offspring. So if I can go to next slide. Punnett squares are going to be your best friend in this case. They are used to predict the offspring with a parent's genotype and their probabilities. So I'll just go through an example question. This should be revision, but in dogs there is a hereditary deafness caused by the recessive gene little d. A kennel owner has a male dog that she wants to use for breeding purposes if possible. The dog is able to hear. So the dominant phenotype or the dominant gene of capital D means that they have normal hearing. Since the dog can hear, this dog will either have capital D, little d as a genotype or capital D as a genotype. What are the two possible genotypes of Gilbert? These two. So anytime they have the dominant trait, they can either be homozygous or heterozygous dominant. There. So you can use those Punnett squares to predict what the genotypes of offspring are going to be, or work out what they are using something called a pedigree chart. So what you see on the screen is indeed a pedigree chart. And a couple of rules on how they work. The first is that squares are for males, circles are for females. And if one of these parts of the pedigree chart is shaded in, that just means that the person is a carrier of a disease. So pedigree charts are used to map how certain disease and certain conditions can be carried throughout a family. And there are a couple of different inheritance patterns that we're going to learn about that you could be tested on. The first one is autosomal dominant inheritance, as you can tell by the title. Autosomal referring to the non-sex chromosomes, so chromosomes 1 to 22. Dominant meaning that the disease phenotype happens if you have any copy of the dominant gene. So if you've got BB or B, capital B's in your genotype. So let's just say an example of this is Huntington's disease, right? It's a disease that causes nerve breakdown over time, but you're not required to know any of that. Usually, if we have a look at our first parent or like the starting parent of the family, we've got a male who is affected by the disease. So they could have either been capital B, or capital B, little b, and a female who isn't affected by the disease. So they're double recessive. And the way that we can figure out what the male is like truly is through doing a Punnett square. And so, sorry, just give me 2 seconds. In order to do a Punnett square, you put one of the parents' phenotypes around the top and one of the parents' phenotypes on the side. Draw a little grid like this, and all of the offspring genotypes are the combinations of the alleles that fall into the grid. So this grid square has a capital letter lowercase. This grid square has lowercase, lowercase. This grid square, capital lowercase. This one, lowercase, lowercase. So if that male was heterozygous, the offspring would be expected to be 50% affected or heterozygous as well, and 50% unaffected or double recessive. What we actually see happening is of the 5 kids that are born, 1, 2, 3 of them are affected and 2 of them are unaffected. So it is pretty close to the expected ratio. If they had 6 kids, maybe it would have evened out to 50%. We don't know. But if we take the alternate option and say that the father was homozygous, I'll just do the Punnett square down here. Homozygous with the recessive mother, and then draw the grid. Every single offspring would be capital B, little b, capital B, little b, capital B little b, or capital B little b. 100% affected. But that is not equal to the actual pedigree chart ratios. So therefore, we can say that the father is indeed heterozygous and not homozygous. And that is an example of how you use Punnett squares to figure out genotypes. So autosomal dominant inheritance means that the dominant allele gets the disease. However, there is another kind of inheritance that is autosomal recessive. So this time around, the diseased individual or the affected individual must contain the homozygous recessive genotype. Now, in this case, every single black square or black shaded shape on the pedigree chart, we can immediately say is double recessive. It's the unaffected individuals that we are a little bit more wary of. So in recessive inheritance, any kind of recessive inheritance, there are these things called carriers that can start coming up, where the parent is unaffected, but they give birth to a child who is affected. When this happens, you have to realize that the parent is automatically heterozygous. Both parents must automatically be heterozygous. And I'll show you why. Heterozygous. So let's just say our gene this time is like R. This parent could be double R capital. They could also be homozygous. Sorry. I'll go RR, RR, and for the recessives. If the parents are both heterozygous, and I do a cross with them, the first offspring phenotype is double R. Then I have two offspring who could be heterozygous, but still dominant, and then 25% chance that the recessive allele gets through. However, if I have a double R situation, and I cross it with, say, heterozygous, because one of them has both copies of the dominant gene, every single one of the offspring will inherit the dominant gene. So none of them will be affected. So the moment that you see this kind of pattern happening where two parents are clean of the disease and they give birth to a child who is born with the disease, that should be like alarm bells ringing that the inheritance type is recessive and that both of the parents have heterozygous alleles. So here, for example, I've got two unaffected parents, one affected child. So this is heterozygous and this is heterozygous as well. Now that I've kind of gone through the whole Punnett square business, the other types of inheritance patterns I'm kind of going to whiz through a little bit. Let me know if you would like further clarification though. Now that we've talked about like autosomal dominance inheritance, we're going to talk about sex-linked inheritance now. This is basically where the gene is on the sex chromosome instead of being in a non-sex chromosome. So X-linked inheritance, because every person is born with at least one X chromosome, this affects everyone. And when you're writing the genotypes of these guys, females will be written as X with either a capital letter as the subscript or X with a recessive letter as the subscript, or and males will be written as X with a capital or lowercase letter, and then Y to say that they have the Y chromosome there. So in X-linked dominance inheritance, an affected individual has the capital letter, has the dominant gene. The recessive gene is unaffected, and then the Y has no effect. So in this case, for example, the mother would be X capital H or X little h, alternatively X capital H, X capital H. And the father is X little h, Y, unaffected with the Y chromosome. And so you can do Punnett squares to figure out what the rest of their genotypes are. If you have a mother who is affected, who gives birth to unaffected children, that is letting you know that the mother is heterozygous. So she still has a copy of that dominant gene that she can pass. Sorry, she still has a copy of the recessive gene that she can pass on to her kids. And paired with the recessive gene on the dad side, some of the kids will be born with no copy of the dominant gene. Whereas if the mother had both copies of the dominant gene, I'll show you what happens. If I've got XH, XH, and then X little h, Y, every single one of the offspring is given a copy of the dominant gene, and therefore their offspring will all be shaded in. But that's not the case here. So we can immediately say that the mother is X big H, X little h, and Punnett squares can be used to find what the remaining genotypes are. X-linked inheritance can also be recessive, so that just means that the affected condition is the recessive allele. And that any mother with X little h, X little h, so homozygous for the recessive allele, or X little hy, a male with a copy of the recessive allele, will be affected. Typically, for X-linked recessive inheritance, males will have the disease more often than females. And the reason that happens is females have the ability to code against the recessive alleles twice. They have 2 copies of the X chromosome, and they only need 1 copy of the dominant allele to be healthy or born without the condition. Whereas for males, they only have 1 chance to code against the diseased allele, and if they do get it, they have to be born with the condition because the Y chromosome has no effect at all. So typically speaking, in X-linked recessive inheritance, the amount of affected individuals, so all of these guys are X little hy, 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 and then this would be X little h, X little h, X little h, X little h, and X little h, X little h. The amount of affected individuals who are male are going to be much higher than the amount of affected individuals who are female. And unfortunately, that's just the way that it is. An example of this is red-green color blindness and also hemophilia. Generally speaking, males are much more the chance of a male developing this disease is much higher than the females. So as example, I'm just going to use this family right here. 2 unaffected individuals. So the male has to be X capital HY. They don't have the recessive diseased gene. The female could be X capital H, X capital H, or X capital H, X little h, and be a carrier for the condition. Immediately, because one of the boys that are born to the parents are born with the disease, the mother has to be a carrier. So if I was to choose the heterozygous genotype, the mother has X capital H, X little h. The father has X big H, Y. And if I do a Punnett square, I get X capital H, X capital H, X capital H, X little h. So 50% of the females 100% of the females are unaffected, thanks to the father. Whereas the males that are born, only 50% of the males will have the dominant gene, and the resulting 50% have the recessive gene. So in this case, the only offspring that can be born with the disease are males with the recessive gene. And you can work out the rest of those genotypes using the Punnett squares. There is another type of inheritance. Obviously, we've done sex-linked inheritance on the X chromosome, but there is also sex-linked inheritance on the Y chromosome. But all of these diseases only affect males because females do not have the Y chromosome in general. So disease only affects people with XY chromosomes. Now, because of this, the only shapes that are going to be shaded in are these squares. And so let's say that this Y-linked inheritance, the recessive allele, is the one that is the disease. I'm only going to give you one example of this because it's not a very commonly assessed question, but the theory is there. So this father would be X little by crossed with X big B, X big B, or X big B, X little b. So here I have all of the males that are born. So X big B, Y, X big B. I'll start with the heterozygous phenotype first. Every single one of the males that are born has the disease gene, and the other combination of the mother's capital B's. Alright, so here you can see that the, oh, sorry, wait. What am I saying? The disease is on the Y gene, so it's X, Y, little b, and then this is just XX. Now, here we can see that every single one of the sons of the father gets that Y gene inherited. So immediately the disease goes to the sons, but the daughters are spared because it's not on the Y gene. So this would be X, Y, little b. This would be XX. These would both be XX, XX. Now this is XY, the healthy copy of the gene. So in this case, it would be the dominant gene. Here's X, Y, little b, XX. Here, because the healthy copy of the gene is the only Y copy of the gene in the entire family, both of the males are born with the healthy copy. And here, both of the males are born with the diseased allele. Like so. This is an XX, and this would be XY little b. Y-linked traits are actually very, very rare. Since the Y chromosome only has 63 genes in general. The way that it works is that if the father has the disease, then any sons that are born to those parents will inherit that condition. Right. So those are the main inheritance linkages. We're going to talk a little bit now about things that are called multiple allele inheritance. In some traits, there are multiple dominant alleles. And the inheritance patterns that result from multiple dominant alleles, one of them is called incomplete dominance, where if an individual inherits both dominant alleles, inherits more than one dominant allele, a new phenotype is expressed. So, for example, if you have a cat species that has a dominant allele for white fur, this is dominant, and a dominant allele for brown fur. Oh, wait, actually, I'll go dominant allele for black fur. This is also dominant. If I was to want a cat with only white fur, they would need the genotype CWCW. This is pure white fur. If I wanted a cat with pure black fur, it would be CBCB. But if I was to get the heterozygous phenotype of CWCB, instead of being, white or black, because both of them are dominant, a new phenotype occurs and the fur color would be gray. Completely different to the phenotypes of the homozygous genotypes. So here, for example, checkered box on a pedigree chart means that they've got both copies of the dominant allele. So this could be CWCB. The mother is also CWCB. And if I do a Punnett square for both of these and cross them, one of the offspring will be born well, 1/4 of the offspring will be born as white cats. 1/4 of the offspring, 50% of the offspring, because there's two of them, will be born as grey cats, and 1/4 of the offspring will be born as black cats, even though both parents started out being grey. And so the whole thing with incomplete dominance is that because there are multiple dominant alleles, a new phenotype occurs when they are heterozygous. Sometimes people say that it's like a blend of effects in the different alleles. And so another example is if you've got a dominant allele for white petals in flowers mixed with a dominant allele for red petals in flowers. Even though it's a heterozygous phenotype, the flower color will be pink, so like a mixture of white and red. So yeah. There is a very similar inheritance pattern to incomplete dominance called codominance. Now, an example of this is blood type, and this was actually assessed in the 2021 paper. So in my external exam, there was a question in paper 2 exactly on blood type. And basically, this has multiple dominant alleles, but also a recessive allele situation as well. Blood types refer to your blood cell's ability to have these proteins on their surface. A-type people have a specific protein on their blood cells. B-type people have a different protein, and then AB people have a combination of the A protein and the B protein. Here, blood types for AB, the alleles for A and B are dominant, but the alleles for the O-type blood are recessive. So we give it like a lowercase I shape. And if the person has both copies of the dominant allele, so if their genotype is IAIB, instead of it being like blood type C, it is a blood type that combines the two alleles together. So you get the protein for A and the protein for B both expressed. A new protein is not expressed. If there was a different protein, then it would be incomplete dominance. But in codominance inheritance, both of the dominant genotypes end up being represented in the phenotype. So anybody with a blood type A can have the genotype IAIA. They can be homozygous for it, or blood type IA little I and be a carrier for the O-type blood. If you have two parents that are both AB blood types and you cross them together, 25% of your kids are going to be type A only. 50% of your kids are going to be type AB. And 25% chance your kids are going to be type B only. And using Punnett squares, you can track what blood types might occur from different kinds of people. And there are some questions that we can look at later on in the lesson that show exactly that. If you want a visual of the difference between codominance inheritance and incomplete dominance, so I've got two flowers here where the parents are white and red. In incomplete dominance, if they get a combination of both of these dominant alleles, the offspring flower will be a blend of the two, so pink. Red and white blend together to make pink. In codominance inheritance, the two dominant alleles share the phenotype, and so there are parts that are red and parts that are white. Hopefully that makes sense. Now, going on to the final topic for the inheritance concept, there is one other multiple allele inheritance pattern called polygenic inheritance, and that is where one characteristic is controlled by two or more genes. This I've given you as the QCEE definition, by the way. And an example of this is human height, sorry, that should have a T on there, or skin colors. Skin color actually is made up of three genes. So there's a dominant gene, A, B, and C, and then recessive genes, little a, b, and c. The more dominant genes you have, the darker your skin tone is, and the more recessive you have, the more light it is. And so this is actually the example of a Punnett square for inheritance with three different types of genes. All of the parent genotypes could be one copy of each of the genes. So either they could be having all like you could inherit all of the dominant genes from your mother and father, or you could inherit all of the recessive genes, or any combination of, two dominant genes and one recessive, or two recessive and one dominant. And basically, once you do a Punnett square, for example, if I was to get the genotype of this individual, you would need to combine the alleles from one of the parents with the other parent, and you would have capital A, capital B, little c, capital C, little c. So they have equal amounts of recessive and dominant here. This would be like a medium skin tone, which you can see from the table. Now, because of this, naturally, human genotypes can code for 7 different kinds of skin tones. The darkest being like completely homozygous dominant, and the lightest being homozygous recessive. And what we call this is you can like map all of these phenotypes on a graph almost. And because there are so many more combinations that yield for like the more middle skin tone, more people are going to be born with the middle skin tone. And if you're able to graph phenotypes like this, we call this like continuous phenotype variation, where there are so many different phenotypes that you can express, you can almost like graph them. Another example of that is hair color. Obviously, lightest hair color being blonde, darkest hair color being black. And there are about, I think it's like 10 or 11 genes that actually influence hair color. So it is a very, very, very continuous gene. We'll just put it like that.